This video will show you how to solve genetics problems using an 8-step outline. Before we get into the outline, I want to say a few words about breeding flowers. Most flowers have both male and female parts. In this particular flower, the anther produces the pollen, which is the male gamete, like sperm. The eggs are housed in the ovary. In the wild, pollen is released from the anther and finds its way to the stigma and descends down the style so that the pollen can unite with the eggs in the ovary to produce seeds. So we have pollen uniting with eggs in the ovary producing seeds. So now along comes Gregor Mendel around 1850, the father of genetics, thinking that he'd like to try an experiment of crossing purple-flowered pea plants with white-flowered pea plants to see what he'd get. So he snipped off the anthers of the purple flower before they matured, so they couldn't produce pollen. Then he went into the other greenhouse and on a paintbrush grabbed some pollen from a white flower plant and brought the pollen back to the stigma of the purple flowered plant. So the seeds that were created contained genes from the white flower and genes from the purple flower. He planted the matured seeds and those plants all had purple flowers. Now, why do you suppose that the offspring of the purple pea plant and the white pea plant all had purple flowers? Let's take a look at what happened. So we know a couple of things. We know that genes occur in pairs and that we surmise that purple must be dominant to white, so we can set up the cross in this manner. First, we add a key. The key tells us several things. That gives us two genes, a big P and a little p. The big P is dominant to the little p. The big P gives us purple colored flowers. The little p gives us white colored flowers. The first step after the key, so it's the second step really, is to identify the parents, phenotypes and phenotypes. The phenotype is their appearance. The genotype is their genetic makeup, shown here. The third step is to identify gametes from each. The law of segregation tells us that the alleles for a character get packed into separate gametes. This happens during meiosis I. Combining the gametes is called fertilization. Fertilization gives us the F1 generation genotype, which is composed of a dominant gene and a recessive gene, a big P and a little p. This results in a purple flower phenotype. So to review to this stage, we have a key, which tells us the two alleles and the dominant and recessive phenotypes of those alleles. Then we have the second step, which is identifying the parent generation, both parents' genotypes and phenotypes. Then we have the third step, which is identifying the gametes from each of the parents. Then we have the fourth step, which is identifying F1 generation, phenotype and genotype as a result of fertilization of the gametes. Okay, next, in order to create a second generation of offspring, the grandkids, we take two of the F1 generations and mate them. So we take two of the purple flower heterozygous, that's big P, little p, individuals, and mate them. So we get eggs that either have a big P or a little p, either a dominant or a recessive. And then we have two possibilities for the sperm as well. Sperm with dominant genes and sperm with recessive alleles. And so using the Punnett square, we see that we get one individual in the top square with two dominant genes. We get two in the second tier down that have a dominant gene and a recessive gene. This condition is called heterozygous. And then in the bottom square, we have a homozygous recessive. So the homozygous dominant in the top square gives us purple flower phenotype. The two in the second tier with the heterozygous genotype gives us the dominant purple phenotype, and the one at the bottom with the homozygous recessive genotype gives us white phenotype. The ratio of phenotypes is 3 purple to 1 white. So to review one more time, we have an 8-step outline. Step 1 is the key. Step 2 is the parent generation phenotype and genotype, appearance and genetic makeup. Third step is the gametes of the P generation parents. Fourth step is the F1 generation, the first filial generation, the kids. Now, step 5 is not really represented in this particular diagram. Step 5 is take 2 of the F1 generations to make them parents of the F2 generation, the grandkids. So you identify the appearance and the genotype of the F1 parents to the F2s. Then you identify the gametes. That's step 6. Step 7 is fertilization. So you combine the gametes in the 4 different squares in the Punnett square and identify the genotypes in the squares and the phenotypes that the genotypes will produce by referring back to the key. The 8th step is ratios. So we do ratios of phenotypes and or genotypes depending on what the question is asking you for.